Hello, let's listen to this one.
Certain phagocytic cells of the immune system, called antigen-presenting cells, seek out foreign cells and engulf them. In addition, despite the best efforts of the immune system, cells can become infected. In both situations, the cell needs a way to communicate with the immune system cells. The phagocyte needs to show what it's captured. The virus-infected cell needs to show that it's infected. Antigen processing and presentation is a way for a cell to display information about its activities.
His Facebook page. Okay, the exciting video continues.
Each helper T cell produces a unique T cell receptor, or TCR, which corresponds to a specific antigen. Each helper T cell also displays a receptor called CD4 on its surface, which recognizes the MHC molecules of antigen-presenting cells. When the helper T cell encounters an antigen-presenting cell with that antigen displayed in an MHC molecule, it signals the antigen-presenting cell to release interleukin-1. The IL-1 stimulates the helper T cell to produce IL-2, as well as IL-2 receptors. The helper T cell can then auto-stimulate itself, keeping it active as it proliferates. Once a helper T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these helper T cells differentiate into long-lived memory T cells, but most mature to produce cytokines to help with the immune response. Helper T cells differentiate into two subpopulations called Th1 and Th2. Th1 cells produce cytokines that primarily activate the cells of cell-mediated immunity, such as macrophages, cytotoxic T cells, and natural killer cells. Th2 cells produce cytokines that activate the B cells involved in humoral immunity.
Activating the B cells. Okay, one more video to explain this, and then we will get down to it.
Antigen-presenting cells, such as macrophages and dendritic cells, have two classes of special proteins that are used to present antigens on their surfaces. Collectively, these proteins are called MHC, which stands for Major Histocompatibility Complex. Class 1 MHC proteins are used to present antigens produced within the cell. A different class of MHC proteins, Class 2 MHC proteins, are used to present antigens engulfed by antigen-presenting cells. Most of the other cells in the body, including infected cells, are also able to present antigens. However, these cells only have...
Okay. Sorry about that. Let's fast forward to here.
Class of MHC proteins are used to present antigens produced within the cell. A different class of MHC proteins, Class 2 MHC proteins, are used to present antigens engulfed by antigen-presenting cells. Most of the other cells in the body, including infected cells, are also able to present antigens. However, these cells only have the Class 1 MHC protein.
Remember, Class 1, all your nucleated cells. Everything except blood cells.
Proteins.
Okay, so let's review this again with the better picture from your book. Okay, so we have a stem cell. These are development of your T cells. In stem cell in the bone marrow, right? It's a T cell, so it goes to school in the thymus, right? It shows up for self-selection class, first day. Does it bind to an MHC? Yes or no? Yes, then it receives a signal to survive. No, sorry, apoptosis, right? Can it bind to an MHC? Yes, you get to go here. So you didn't bind to yourself, so you get to move on. Now, can you receive a signal? Can you recognize an antigen on an MHC? If you can't, you don't necessarily have to be apoptosis right away. You can just kind of go and be an immature T cell. But remember, when you finally bind to your antigen, you become an activated T cell, right? If you can't, you get apoptosis. If you can, right, now you're a regulatory T cell. Yes? Does this make sense? Do you remember this? Again, found in the spleen, lymph nodes, and MALT, small percentage of B cells. Oh, I'm sorry. This is T cells. Sorry about that. T cells. Clonal deletion of T cells. Vital that the immune response not be directed against autoantigens. So you first have to go to selection school to make sure that you're not attacking yourself. Non-self selection school. If you're reactive to yourself, your body eliminates you. You're apoptosis. Lymphocytes that react to autoantigens, aka self cells, undergo apoptosis. Sorry, that's the first class you get to take. You don't pass that, you die. Clonal deletion of T cells, so immature T cells undergo 4 potential things could happen to you. T cells that do not recognize your MHC protein, also gotta go. T cells that recognize yourself, also gotta go. Some self-recognizing T cells become regulatory, right? Cruise of the lymph. Then cells that actually recognize their MHC protein and foreign epitopes become protective T cells. Okay, and that's exactly what this picture is showing you. B cells, found in the spleen, lymph node, and MALT. Sorry, I had that. Now we're going to show a picture of the B cells. There's not a huge amount of these B cells circulating in your blood. Major function is the secretion of antibodies. Specifically, of the B cell receptors. Each B lymphocyte has multiple copies of its BCR or B cell receptor. So each of it has a bunch of receptors. Remember, that was, when you went to B lymphocyte school, you had to pick a receptor. You can only recognize one epitope, and the entire repertoire of individuals in your B cell receptor is capable of recognizing millions of different epitopes. So here's what that looks like. You have a little epitope here. Here, this is a cytoplasm. This is your cell membrane, just to orient you. And then here's some sulfide bonds in between your antigen binding sites, and there's an epitope from an antigen. When you finally bind, you become an activated B cell. Yay! Genes for constant and varying regions of the B recognition site occur at discrete loci. And what else do we want to know about that? Antibodies are immunoglobulins. They're secreted by activated B cells called plasma cells. Do you remember we talked about that? We said, what do your plasma cells do? And I was like, your plasma cells go crazy and like activate all these antibodies. Remember that? So this is what's happening. You've got an activated B cell. It can become a memory cell or a plasma cell, right? It can go do 2 things. It becomes a memory cell, then it just goes and remembers so it can activate the troops next time. If it becomes a plasma cell, it just goes nuts and produces antibodies like crazy. Okay? So that's what happens when it becomes activated. That is what it looks like, the actual structure of it. Okay. Antibody function. So again, the antigen binding sites are complementary to epitopes. Activation of what happens when these antibodies from your plasma cell gets released, right? You have opsonization, which is it pokes holes in it. Basically it dies. Activation of complement and inflammation, again, is happening. Remember complement from last time. You don't have to know all the steps of it, just know complement happens, and it tends to cause inflammation, and that is your antibody antigen function. It can neutralize it. It can coagulate, like just kind of clump all together. Optonization. See how nice that is? It popped a hole in it. That's exactly what it did there. And we are not probably going to listen to any of the rest of these videos. I think this is probably enough. There's classes of antibodies. You do not need to know those. Here's, again, the clonal deletion of B cells. So you've got a stem cell going to school, right? Here's all the B cells. If it binds with self, it gets apoptosed. If it binds with an antigen, it gets sent into the blood vessel, into the spleen, and to cruise around and look for its little antigen that it's looking for. Okay. Cytokines are secreted by various leukocytes, and they complete signals. And I think that's really, you don't have to know all these things. These are just signals. And that is basically all that we're going to go over for this chapter. you need to put it all together yourself and send me your